Assalamu alaikum and hello everybody. Central nervous system depressant topic is the next lecture, which includes benzodiazepines and anticonvulsants or anti-epileptics in this lecture, and antipsychotics in the next lecture. So I'll start now with the benzodiazepines in this part and anticonvulsants in the second part. These are the learning outcomes of benzodiazepines, and I'll talk about anxiety because benzodiazepines are the main agents used for the treatment of anxiety. Then I'll talk about GABA receptors and explain the mechanism of action of benzodiazepines. Then we'll explain the structure-activity relationship uses and give examples for these agents. To define anxiety, which is your reaction to stress, worries, and fears. So it is like sense of apprehensive expectation, or in Arabic الإحساس بالتوقعات المخيفة. And anxiety disorders can be classified into, for example, specific phobias, chronic high level of worry, social phobia, and others, as you can see in this list. The three main neurotransmitters associated with anxiety are first, GABA or gamma aminobutyric acid, which is the major inhibitory neurotransmitter in the brain. Second, noradrenaline, and third, serotonin or 5-hydroxytryptamine 5-HT. Actually, GABA neurotransmitter works on GABA receptors, which are classified into first, GABA A receptor, which is the ligand-gated chloride channel or linked to chloride ion channels, and GABA B receptor or G-protein coupled receptors, which are activated by GABA, and the last GABA receptor is GABA C, which is also a ligand-gated chloride channel. So both GABA A and GABA C are ligand-gated chloride channels, so I'll mention only GABA A in the next slide. So I'll talk about GABA A receptor, which is the most common or prevalent in mammals and located in the brain, and usually deals with emotions and memory and linked to chloride ion channels, as I mentioned before. And the activation of this receptor can lead to increase the concentration and the influx of chloride ions. Benzodiazepines, which are the main topic in the first part of this lecture, are the drugs or agents usually target GABA A receptor. Example of these agents is diazepam or Valium brand, which is very common in clinical practice. And the structure of diazepam is shown here in this slide, and you are not expected to know it or memorize it. This scheme is showing the three binding sites of GABA A receptor, which are first, the GABA site, second, the benzodiazepine site, and third, the barbiturate site. And actually, barbiturates are also CNS depressants and sedative hypnotics. Activation of this receptor leads to an increased influx of chloride ions through the cell membrane from outside to inside the cell, and give the fast inhibitory synaptic transmission in CNS, as you will be or maybe already covered this in pharmacology. Benzodiazepine structure is shown at the top, and barbiturates are examples of drugs work on GABA A receptor, while baclofen or parachlorophenyl GABA, which is antispasmodic or agent suppress the muscle spasms, is an example of drugs work on GABA B receptor. Here are some examples of benzodiazepine agents with the most related medical condition. For example, diazepam or Valium brand, alprazolam or Xanax, and lorazepam agents are used to treat anxiety, while nitrazepam and temazepam agents are used as hypnotics or sedative agents, and clonazepam and also diazepam can be used as anti-epileptics, and midazolam is used as basal anesthetic agent. Some undesirable effects caused by benzodiazepines, for instance, and in addition to sedative effects, benzodiazepines can cause confusion, amnesia or dementia, which affect the memory, and also can cause ataxia or lack of voluntary coordination of muscle movements. And also these agents can cause tolerance or dependence, especially when long-acting benzodiazepines are used. So the short-acting agents are preferable for short-term treatment when required. In terms of pharmacokinetic, actually the rate of absorption and the existence of active metabolites and the lipophilicity are important factors in determining the therapeutic uses of benzodiazepines. For example, a lipophilic agent with rapid absorption and lack of active metabolites can be used as hypnotic, while the agent with the opposite characteristics is used as anxiolytic agent. As I mentioned before, benzodiazepines work on GABA I receptor, which is linked to a chloride channel, and activate this receptor or this channel and lead to an increased influx of chloride ions into the cell. However, benzodiazepines are not directly affecting the channel, but can modulate or alter the GABA site on the benzodiazepine receptor and activate the chloride channel and give the pharmacological action. In relation to the structure-activity relationship, SAR, of benzodiazepines, actually a huge number of these drugs are synthesized, but only limited number of them are considered and used in a clinical practice, with the most promising features available can facilitate the binding of these drugs into the targeted receptors and give their action. You don't need to memorize the structure of benzodiazepine, but you do need to know the numbering system when you are giving the structure in the exam as shown in this slide. So numbering order starts with the nitrogen at position 1, then number 2 of carbonyl group, and so on, as you can see in front of you. This is the longest and the most important slide in this lecture. So there are 6 important points to be mentioned here in the benzodiazepine structure, which consists of benzene rings. So the name of the class starts with benzo, and one of them, shown at the top left, it's used into the 7-membered ring, contains 2 nitrogens, so we call it diaza, and this 7-membered ring is unsaturated and contains double bond, so it's ethyne, and the whole 7-membered ring called diazepine, and the whole structure is called benzodiazepine. The first point in the SAR of benzodiazepine is the NR group at position 1 at the top, which is the optimal and essential for activity, and R group can be varied, for example, hydrogen in lorazepam, and methyl or CH3 in diazepam, and so on, as shown in the next slide. The second point is the aromatic ring A, which is more active than heteroaromatic ring, which contains heteroatom, for example, oxygen, nitrogen, or sulfur. The third point is the presence of electron withdrawn group at position 7, as you can see in this slide, which increases the activity of the drug by increasing the electronegativity and pulling the electron density from the benzene ring towards itself or towards this group. For example, removing the electron density from the pi system in the benzene ring and make the ring less reactive, and therefore the electron withdrawn group is called deactivating group. Examples of these groups are halogens, cyano group, and carbonyl group. Also, adding electron withdrawn group make it more difficult to introduce new groups onto the benzene ring, and also good for the stability of this ring. The fourth point in the SAR of benzodiazepine structure is the phenyl group at position 5 or ring C at the bottom of the structure, which increases the activity, especially when electron with the wrong group is present at position 2 dash or positions 2 dash and 6 dash. However, substitution at position 4 will decrease the activity. The fifth point is the substitution with or without OH group at position 3. So, if no substitution or introducing of polar group or hydrophilic group with OH at position 3 will increase the rate of phase 2 metabolism reaction and make it easy to conjugate and excrete it rapidly with glucuronide conjugation. So the duration of drug action will be short. And an example is the lorazepam drug in the next slide with polar group or OH at position 3 make it short or intermediate action agent. However, if nonpolar or hydrophobic group was introduced at position 3 will increase the rate of phase 1 metabolism reaction and the active metabolite resulted by hepatic oxidation in the liver will increase the duration of the drug action. Finally, the sixth point is related to introducing of hydrogen acceptor group like oxygen or nitrogen at position 2. So here we have the oxygen of the carbonyl group at the top of the structure, which acts as hydrogen acceptor and increases the activity of the drug. Also, introducing of imidazole or triazole ring shown at the bottom right of this slide onto the positions 1 and 2 at the top and fused to the diazepine ring at positions 1 and 2. This also will increase the activity of the drug. Here are examples of generic and brand benzodiazepine drugs, and again these structures are not expected to be known by heart, but you must know the important features of these drugs as I mentioned in details in the previous slide. Now we'll move to the second part of this lecture, which is a relatively short part and based on the anticonvulsants or anti-epileptic drugs. These are the learning outcomes and I'll mention to them as we go through the lecture. As you know, epilepsy is a consequence of sudden and excessive or too much firing or generating of neurons. Epilepsy can be classified into partial, which can be either simple or complex and cause impairing of consciousness أو ضعف الوعي. This class of epilepsy can be developed to the second class, which is the generalized epilepsy and categorized by absence from 2 up to 30 seconds and commonly takes place in children. Also, generalized epilepsy can be myoclonic, which corresponds to shoulder shaking, or can be tonic-clonic, which categorized by laying down with a stiffness, and this situation could be emergency if the epilepsy takes more than 5 minutes. Also, there is another type of epilepsy, which is unclassified and less common than the first two types above. This is an important slide and explains the mechanism of action of the anti-epileptic drugs according to their targeted site. For example, phenytoin, carbamazepine, lamotrigine, and others are agents target the sodium or calcium channels, so they called modulators of sodium or calcium channels. However, thiagabine and vigabatrin agents target the GABA system, and thiagabine stops or reduces the reuptake of GABA, while vigabatrin blocks the GABA aminotransferase enzyme, and this provides more available GABA to bind to the receptor and slow the electrical signal. Also, felbamate is another anti-epileptic drug which inhibits the N-methyl D-aspartate or NMDA receptor, which is the glutamate receptor, and gives the action. Full details of the mechanism of action of the most important and common anti-epileptic drugs, phenytoin, carbamazepine, and lamotrigine, are shown in the next slide. So, as I said before, the phenytoin, carbamazepine, lamotrigine, and also phenobarbital target the sodium or calcium channels. And just to bring you back to your physiology notes, and as you know, after each depolarization, sodium channels remain in inactive state for a period of time, and as these channels are blocked, so number of neurons are minimized and decreased, and consequently the spread or transfer of electrical seizures to the adjacent or to the next brain region are reduced or stopped. So keeping this situation or keeping this inactive state is the mechanism of action of these drugs. Finally, here are the critical structure elements of the sodium channel blockers or the drugs mentioned in the last slide and shown in this slide. So the important elements or characteristics of these drugs are shown in the dotted rectangle, and these three elements are: first, the hydrophobic unit R or phenyl ring, and you can see the R capital letter inside the phenyl ring. And the second element is the electron donor group D, also shown in these structures and indicated by oxygen and nitrogen atoms, which are good electron donors as they have lone pair or pairs of electrons, can act as bases in acid-base reactions. The third element is the hydrogen acceptor donor group HAD, assigned by amine or amide groups in the structures ahead. So these three elements are important and essential for activity. So after listening and well studying of this lecture, you should be familiar with the outcome listed in this slide. Thank you very much for your listening and attention.